new to the group and are not on the mailing list, we have a Google Groups mailing list, VGP Assembly. If Camilla or Marcella could send me a list of email addresses for all of the students who are joining, we'll welcome you to our Friday calls, which we use to do progress updates and answer any technical questions and so on. So welcome. We're excited to have you all here, and I'll hand it over to Arang, I guess, is beginning the presentation.
Okay, I'll try to share my screen. All right. So, all right, welcome everyone. So here I will give everyone a brief introduction about the VGP standard 1.5 pipeline. My name is Arang Ri. You probably got an email from me either via DNA Nexus or directly from the VGP Assembly Google Group mailing list. I'm gonna share the slides after this orientation, the training is over, so don't worry about that. So here, I just wanted to give a brief introduction about the VGP Assembly Working Group. Adam is the chair, he just gave an introduction. And my name is Arang Ri, and Eric, Richard's group, Jean's group, Zeman's group, Kirsten's group, Harris group, Goji's group are all attending on a regular Friday basis assembly group call. And so let me get back to this. So what we basically do is we collect data from the genomes that we want to build the reference genome, and we store them, we archive all the data on a public data source, which is provided by Amazon Web Service. They provided us a public storage so we can put everything there. And then we go through the pipeline, and the pipeline can be run either locally and also on the DNA Nexus platform, which Maria will introduce more later. And with that, we get an assembly. And our assembly working group, we are mainly focusing on designing and evaluating assembly pipelines. We not only do the assembly, we also do the curation and also submit them to the NCBI and EBI, so everyone can use that later for a reference genome. And besides, we also collect and organize the data and continue finding new methods on how we can generate better assemblies. Okay, let me get back. So the goal here is to make good reference quality assemblies. So initially we had a standard, which is a goal that we wanted to capture all the genes properly, correctly in their form, and not only the genes, also the structure in the genome. We had some numerical goals set up here, which is likely to change in the near future. But it's essentially about the contiguity. We want to build sequences, gapless sequences, as long as possible with high base pair accuracy, and also want to reserve both haplotypes, which I will introduce a bit later. And also want to build the contigs into scaffolds, into higher order scaffolds, which will be closer to the chromosomes. Okay, so the data flow, I added this slide, especially for this training purpose, so that everyone understands what's going on behind the scenes. So we put the data, as soon as we generate the sequencing data, the sequencing centers or the researcher who did the genome sequencing is uploading the data on the Genome Arc, which is on the AWS bucket. And from there, we symlink the data to the DNA Nexus platform. And as we generate the data, we copy them back to the GenomeArk and generate the symlinks. And all the computational work via DNA Nexus platform is done on the EC2, the cloud-based web source on the Amazon Web Service.
Can I just mention, Arang, the reason for doing that data shuffling is because we have to pay for data storage on DNA Nexus for gigabytes per unit time that they're stored, whereas Amazon has provided free data bucket for us that includes free ingress and egress. And so at all times, we want to store the bulk of our data on that GenomeArk S3 bucket because it's free of charge compared to the things at DNA Nexus.
This is Eric. I'll just add that it was, yeah, so I want to thank DNA Nexus, that was the one that negotiated the free storage with Amazon.
I didn't know that. All right, so yeah, so there are two ways to access the data. One is downloading directly via the DNA Nexus platform, which I believe Maria will tell you later. The other way is to directly access through the Amazon command line interface. Yesterday night, I uploaded a brief README introduction on how to install the AWS CLI and get started with downloading and retrieving what's in the bucket. So I hope I heavily encourage everyone here, just try to follow what's in the instructions and try to list what's in the Hummingbird bucket, for example. And so this is how the GenomeArk storage looks like. So it starts with S3 GenomeArk species. Under the species, there goes the species name, which is the ordinal level name, the class name, and the species name. And below that, we assign each genomes a unique ID. So as you can guess, the first three letters here comes from the calypt part, and the three letters come from the species name. And up here, this is either B for birds, M for mammals, and A for amphibians, R for reptiles. What else?
F for fish.
Oh yeah, thanks. Yeah, so under each genome, we have the genomic data. And at the same level, we also have the assembly intermediates. And there's another level for the transcriptomic data as well. But here I just put the genomic data and the assembly intermediate. So when you list what's under the genomic data, you can see each platforms here, and under each platforms, there are the raw data stored. So all the data will be linked to the DNA Nexus platform in the same structure. And as you start running each pipeline apps, you will start generate data. And I encourage you to follow this structure, making this folder. Under this, build the intermediates, and under this, for example, if you're starting with Falcon Unzip, generate a folder Falcon Unzip and put everything under this directory. Because the way we are copying back is mirroring what's in the DNA Nexus platform. So it will automatically follow all the data structure, what's generated in the DNA Nexus project. Yeah, so as you can see under intermediates, I highlighted it in bold because this is the most important directory that most of you will be uploaded into this folder. There will be essentially one directory per tool, and there will be one FASTA file that's been generated from each tools. And there are as well some intermediate directories as well. If for those tools that we only need the alignments, there might be another directory here. Here I just put everything that generates a FASTA file. And this is how the pipeline looks like. As I've mentioned, we start with the PacBio raw data, and so all the raw data are available. Let's say are all available on the DNAxis project. So you start using the PacBio reads, running Falcon unzip, and one round of Arrow, which builds the contigs, the sequences without the gaps, and polish it with the reads that's used for the assembly. From here, we get the primary contigs and the alternate haplotics. And after that, we go through another step, only using the primary contigs. We go through the purge haplotics. Here, we align all the PacBio row reads back to the primary contigs only, and try to deduplicate those that should go to the alternate haplotics but are left here. I will cover that a little later. And from here, once we have gone through the purge haplotics, you get two FASTA files, one the curated FASTA files, one the purged haplotics FASTA file. So those, the purged haplotics will go as P2 and will get combined with the C2 for later purposes. We only use the primary contigs, the curated primary contigs for further scaffolding, so we are not getting confused by the other haplotypic sequences. So starting with the P1 contigs, we go through the SCAF10X. Oops, what happened? Sorry for that. My PowerPoint just died. Let me. Okay, sorry for that. So yeah, so after we get the curated primary contigs, we go through 2 rounds of scaffolding using the 10x reads. Here again, we align all the 10x reads to the primary contigs and try to find the contigs order and orientation that belongs next to each other. And from there, we do another round of scaffolding with BioNano and another round with Hi-C. It's similar as we do the 10x scaffolding. And here, all the names are the abbreviations that are used for generating the intermediate FASTA files for abbreviation. After we finish the scaffolding with Hi-C, we go through 3 more rounds of polishing. We go again with 1 more round of Arrow polishing. Here we align all the reads to both the generated S3 scaffolds and the alternate haplotypes, the Q2 contigs. We align all the reads back to both primary and the alts, so we can polish both, and also close some gaps that may be able to closable by the PacBio reads. The gaps that were inserted by either 10x, BioNano, or Hi-C. And then we go through 2 more rounds of polishing using the 10x reads. It's basically similar to how we do the variant calling of the reads and of the reference assembly. So here, the difference is that all the variants that's called as homozygous means that all the bases and that site does not agree with the assembly, which means the Illumina reads does not agree to the assembly. So those will get replaced by the reads suggested by the Illumina reads. And the final T3 assembly will get renamed to pry.asm and alt.asm, and we'll send to the Sanger team and Harris's team for doing another round of curation. So if anyone has a question, please feel free to interrupt anytime. So here I will, at the rest, I will briefly introduce what's behind the logics. So here, as I have described in the pipeline, we do the contigging first with PacBio reads, which the read length is about 10 to 50 kilobase pair long. So starting from here, we do iterative scaffolding using the platforms that give longer linking distances. So here the 10x read molecular length distance is about the range of 50 kilobase pairs to 100 kilobase pairs. And then we go through the bionano scaffolding, which the CMAPs, the in silico CMAPs, not the in silico, the bionano molecule CMAPs are around a meg or 2 megs. And after that, we do the final scaffolding step with the Hi-C reads, which covers most of the genome in approximate lengths close to, so here the interaction distance is almost the size as the chromosome length.
Ron, quick question. On this PacBio read length, the range, could you just quickly say, I don't think in the starting assembly, everything that's like 100 base pairs to 10,000 or so goes in. Only reads of above certain size go in. Is that correct?
Yeah, that's correct. That's only for detecting the overlaps. But for building the assembly, I believe all the reads are used. I think the reads above at least 1 KB are used, I think.
I think the default is 10,000 base pairs, but you can set it to lower.
Yeah,
that's right. And the shorter reads are used to error correct the longer reads in the assembly typically.
So is that a choice in the Falcon Unzip or the Falcon software here? Is that, do you get to set those parameters?
Yeah, so Maria, I think Maria will cover that too. There's an option in Falcon Unzip.
Okay.
Yeah, most of the parameters have been streamlined, but you can go back and set some of those parameters manually as well.
And if somebody sets it manually, does that change it for everybody or just that person's account?
Just that person's account.
Okay.
So here, the default parameters are chosen to get the maximum contiguity and get better contiguity results. So it does not mean that it gives you all that's in your genome. It's there. We have recently found that some part of the genomes could get missed because of those short reads. Yeah, but so I would first encourage people to use the default parameters, because changing the parameters might cause some unexpected results.
Yeah, I agree.
Yeah, and for comparing assemblies to assemblies, it's better to have assemblies built with the same pipe options parameters. So yep, I believe the next one is about the repeats. So here, these were the genomes from the first batch of release of the phase 1 genomes. So here the black bars are the contig and 50s in megabases. But as you can see, some genomes get pretty good contiguities, but some genomes very low, like the SCATE and the channel Blenny. And we can see that each genome has a different repeat fraction. So the contiguity is affected by various factors, but here I believe that the highest factor that's causing fragmentation is the repeat contents in the genome. So each of you in this session will get assigned a genome after the training is over. And you might get surprised that your genome might not have such a good contiguity. So here I'm just warning you that you might get something that looks very good, or you might get something that looks surprised. Don't be too much surprised, and just communicate with me or with Marcella or others in the VGP assembly group. And the other factor here we found in Falcon Unzip was, so here the Falcon Unzip generates two assembly sets, two contig sets, the primary set and the alternates. The primary set here is mostly containing one haplotype and the homozygous part of the genome. And the alternates get split out during the unzipping process. But sometimes if the genome is too much diverse, which means the two sister chromosomes are highly different, Falcon detects them as unique part of the genomes and puts them in the primary sets. Or sometimes the alternates, yeah. So here, sometimes the thing that should go to the primary set goes to the thing that should go to the alternate set goes to sorry. So if the genome is too much highly divergent, it detects them as different part of the genomes, and those parts get sometimes go to the alternate sets, which should belong to the primary set. So here we found that some amount of genome that were assembled as primary contigs should actually go to the other haplotype. And that's why we do the purge haplotics. Here, the SCATE and the zebrafinch female genome were the most highly heterozygous genomes. So here, the expected, you can see that the primary contig size, these are the sum of all the primary contigs. You can see here that the sum is going above a lot more than the expected genome size. And so here we run purge haplotics to deduplicate those contigs. And I just added this slide to give you an idea what you will expect by running each scaffolding steps. So here, the C1 is the Falcon Unzip contigs, one round arrow polished. The S1 is the one with SCAF 10X. So here you can see the contig N50s, which is a normalized average, similar to the average, goes higher and higher as you go through rounds of scaffolding. And after you get back the scaffolding results from the curation team, you might get the N50s back slightly lower, which means that the scaffolding steps might introduce some misassemblies, misjoins, and got broken back by the curation team. I don't know, is everyone familiar with the sequencing platforms? I guess, I can, only a few people in this.
You mean the data type?
She means like differences between PacBio, BioNano, Hi-C.
Mm-hmm.
That's what she has down here, like the Hi-C on the right.
Right.
Yeah.
Yeah. So here I just want you to keep in mind that the 10X molecule reads are Illumina, basically Illumina Paradigm reads with barcodes labeled. So all the reads sharing the same barcodes are assumed to belonging to the same genomic locus. So it's a mapping-based scaffolding. And for the BioNano CMAPs, it's an optical map which labels 5 bases of the genome. It's a 5-base pair pattern of the genome, and labels them and then measures the intervals between each labels. This is more likely an approximate measurement and gets transformed to digital, like approximate base pair numbers. And usually the molecule length is pretty long. We have seen recently with the DLE enzyme that it goes almost to the chromosomal arm size in some genomes, if you are lucky. And the Hi-C is more like an approximate method. It tries to bond the genome as they are in the cell. And so everything that's close has a higher probability to get bonded together. So here it's also Illumina sequencing, paradigm sequencing based method. So each pair generated after the bonding and ligating and cutting represents one interaction that got caught by the bonding. Right, so yeah, I think.
Aran, can I ask something?
Yep.
Camila here, hi.
Mm-hmm.
For the Hi-C, the two sides, so the read 1 and read 2, do they necessarily correspond to the two different parts of the structure? Or you can have the break anywhere?
So the break can be anywhere.
I believe the reads are filtered to just the breakpoint reads for the scaffolding.
Right. So some reads detect the restriction enzyme site, which is here in this picture. This part here is the restriction enzyme site. You can see that the two molecules are cross-linked at this point, and most of the cases the two read pairs are coming from two different molecules, but in some cases a read goes through this restriction enzyme site, and if you're unlucky, you get just one molecule sequenced in paired end, which is the same as Illumina whole genome sequencing. So here, during this Hi-C scaffolding, we try to filter and leave the read pairs left that contain at least one restriction enzyme between the paired end.
And back to Camilla's question, the current Hi-C protocol that we're using labels these junctions with biotin and does a pull down on the wet lab side. And so the fragments that you end up sequencing with Illumina have been enriched in the wet lab for the actual junction sequences. So the majority of them should be sampling real interactions between the molecules, but you do get some fraction that aren't.
Okay. But you can potentially recognize by identifying the restriction enzyme sites.
That, and when you map the paired ends back to the contigs, if the two reads are essentially, right next to each other on the contig, then it's not an informative pair for scaffolding.
Okay, okay, thanks.
Right, so I think this was my last slide. Oh, yeah. So I believe that I could go do another training about the evaluation, but I think this will be more helpful after we have something to evaluate. So here I will just give you a brief introduction, what we are doing at the curation step. So the GEVAL team at the Sanger are aligning all the raw data types back to the assembly and see if there are some alignment weirdness going on and detect misassemblies. And they also do, what's it called? They also scan whether there are some contaminations there and try to get rid of those sequences. And with evolutionary highway, it's a chromosomal synteny-based analysis. Try to see compare with the closest available genome assembly or reference genomes, whether the genome has the expected chromosomal synteny or not. Here, we do not have the correct answer, but still it's helpful in case we know that this genome is a very closely related species to the other species that's available. And another approaches are using the k-mers to see whether we have the sequences completely captured. And similar to that, we also use the BUSCO-stats, which is a set of genes that are known to be universally orthogonally maintained as a single copy in a genome. Here we measure how many genes we captured, how many are complete, how many are fragmented, how many are duplicated. And we also measure the base level identity by aligning back the 10x reads and calling the variations. And if other data are available, we do FISH or genetic maps or linkage disequilibrium based analysis for further scaffolding correction. And we have a GitHub repo for the pipeline. If you're interested to learn more what's going on the, I've seen Maria also posted all the app sources on the GitHub. And here, when you go under the pipeline, these were the scripts that I ran locally for the first data release. So I guess you can try to run it locally if you are interested as well. And that's it. I will also invite everyone to our VGP Slack channel. I'll send an invitation after this training session is over as well, so we can discuss if you have any questions there. Right.
Arang, let me say that, I mean, it's a lot to take in, but I also told them already that we'll be here, so we can go through each step as they are like running it, and we can remind all of the steps and what's going on. So I'll be around, and we can go step by step as soon as they are running the first genomes. It's no problem. And the other thing that I was thinking, some like K-MER analysis to see the composition of the 10x reads prior to the assembly would be nice, right? So we can like see again the estimated genome size and the heterozygosity, because knowing the heterozygosity prior, we will also expect some, like, we'll know how optimistic we can be about the fragmentation of the contigs and things, right?
Right. Yeah. So for doing that, we use GenomeScope. I use GenomeScope on my end using the intermediate files from SCAF10x. It has a step where it trims out all the barcodes at the beginning of the read one pair. So I'm just using those reads for generating the GenomeScope based analysis. So yeah, I think it will be very helpful if you could start doing that before doing the assembly or while the assembly is running to understand your genome.
Yeah, sure.
And we do have that tool as an app on the platform as well, so I'll show you how to run that.
Okay, good.
Yeah, and there, in Falcon Unzip, Sergey planted GenomeScope there as well. So it's also, it can be found in the intermediate files in the Falcon step. It's generated after we get the corrected reads. So I think it was under stage 2 or so. There will be a folder generated named GenomeScope. So you can see the genome.
For the Falcon corrected reads, you mean?
From the Falcon corrected reads as well.
So how do you suggest that we do this in practical terms? So we have the students here, we have 3 students for now. I can even organize more students, but I prefer to start small, to see how it's gonna work. And so the question is, should they get one genome, each one genome? You're gonna send us links or the name of the species, and then they can go directly there. How do you see this working?
So in the next half of the presentation, Maria will walk through practically how each of these assemblies can be generated. As for how we assign who will do what, I envision that we will handle that on our Friday calls. And we can get an update from the sequencing groups about what genomes are available and have all 4 technologies done and are ready to be assembled. And then at that point, we can assign ready to go genomes to students that are able to assemble them. So I imagine we'll handle that on a week-by-week basis on the Friday assembly calls.
Okay, sounds good.
And yeah, I think one genome per student makes sense in the near term.
I would also encourage all of the students to join the Slack channel so that they can ask questions as they're doing the assembly.
And I have also a question for the default parameter.
For the default parameters.
Yeah, for the assembly, as I heard you from you, you suggested some. Please use the default parameter when we sequence, when we generate the assembly. So, is there any recommended option to control lineage, such as a mammal or fishy or reptile?
To control lineages. So far, we haven't made the parameters available specifically to lineages. So all genomes, regardless of what lineage are, they go through the same pipeline. There are some special cases, but that's not lineage based. It's more like if your assembly looks too terrible with the standard pipeline, we do some variation. If you have parents available, we do the trio binning. So we can split out the reads and assemble the genomes per haplotype. But that's using Canoe, not Falcon, unzip. And yeah, and this gate is also gone through another assembly process other than Falcon unzip, which had only like less than a meg and 50 contiguity.
Thank you.
Mm-hmm.
I see there's also a question from Yuri.
Yeah, can you hear me?
Yeah.
Yep.
Arang mentioned that some parts of the genome might be missed. I wonder what they are, and is there a way to bring them back in?
So for that, we are still investigating. The easy way to think is if you know which sequences you are missing, you can fish them out from the row reads and see what happened.
Mm-hmm. Okay.
Yeah, but because we are dealing with genomes which have never been assembled before, and there is no reference, so here the version 1 standard pipeline, we tried to generate the first draft with the best quality that we can generate with the existing methods.
So how do you even know that you are missing something?
We have some groups who are highly interested in some genomes, let's say the platypus.
Mm-hmm.
Doji Zhang's group from BGI, they are highly investigating, and they had their previous assembly built from Illumina short reads, and they compared their genome to their highly curated Illumina-based assembly and found some part of the genomes were missed.
Okay.
I think
So there are some whole genome approaches to do this as well. The typical way is that we get a genome size estimate, either from the Illumina data or from past work with flow cytometry, and compare the assembled size versus the genome size, and that can uncover any kind of large systematic issues with the assembly. It can be much harder to spot a single gene that's left out, but this also is not uncommon, and gaps within reference assemblies have existed since assemblies were invented. So we try to spot any large systematic errors using looking at the data and doing validation approaches that we have. The individual genes are left up to the investigators. If there's a gene that they're interested in that's missing, it's usually in a gap within a scaffold, and we can go back and investigate if it's able to reconstruct that. The two primary culprits in the past have been regions of high heterozygosity, which can confuse the assembler, as well as high or low GC regions that have unusual sequence composition. That's less of a problem now with PacBio than it has been for Illumina, but that's just an example of a typical issue.
Okay, thanks.
And
Yeah, go ahead.
Right. I have a conceptual question. When you talk about repeats, do you mean the low complexity regions that are highly repetitive, or any part of the genome that might be duplicated is also considered a repeat?
It can be both of those cases. There's kind of two categories of repeats I usually think about. Local repeats, which are these kind of low complexity, usually short tandem repeat sequences or satellite DNAs. And then the global repeats, which would be your segmental duplications, or your 5-10 copy, couple KB type duplication. Typically in the genomes that we're looking at for these vertebrates, a lot of times those will be transposable elements or old LTR elements, kind of ancient viral integrations, and those can be on the scale from 500 to 20,000 base pairs and duplicated in varying quantities depending on what species you're looking at. And that really, when you saw Arang's plot of the repetitiveness of the various genomes, and some things like the skate and so on appear more repetitive than others, that's usually due to an explosion of kind of mobile elements in that genome relative to the other genomes. And you can usually see that in the genome sizes as well. Larger the genome size, the higher chance that you have a lot of these duplicated elements. And so both those short tandem low complexity sequences and these kind of scattered transposable elements cause most of the issues.
Okay. Do you think the pipeline can handle well the whole genome duplications that happened in some fish, for example?
Typically, those are ancient enough that they've diverged enough that you can separate them out. Usually our rule of thumb is if sequences are more diverged than, say, 2 to 3%, the assembler can tease them apart without issue. And so when you think about repeats, you need to think about the length of the repeat. In the case of a whole genome, it's going to be the whole genome as the length, but also the divergence of the repeats. And so it's the more recent duplications that are under, 0.5% diverged that are the most difficult to resolve. And whole genome dupes, typically it's a few percent divergence, and those you can assemble out separately.
Okay.
Are there any more questions? In that case, I'm going to start the platform demonstration portion of today's demo. Again, feel free to jump in with any questions as they come up. So I'm going to share my screen here. So what you'll see here is the DNA Nexus platform login page. I think most of you have created accounts already, so you've at least gotten this far. If you haven't made an account, go ahead and make one now by going to platform.dnanexus.com and going to this create account button, and then put it in the chat so that Arang can add you to the VGP orgs or groups. And then I should point out that I'm just going to be focusing on the web UI portion of the platform, but everything I do on the web UI, you can also do on the command line using our command line client. So for production, that might be easier for running jobs at scale. So I'm going to go ahead and log into my VGP account. What you'll see when you log into the VGP account is a whole bunch of projects that have been shared with the VGP organization that have all of the species identifiers. And then you'll also should be able to see this VGP tools project that has the pipeline components. Before I go really into details about the pipeline, I just want to point out a couple of resources that I can't actually access because the screen sharing thing is in the way. So just a couple of tabs that would be useful, which is on the VGP GitHub page. I added a scaffolding workflow README that has an overview step-by-step of the scaffolding workflow because it's pretty complicated at this point. So this just tells you step-by-step what you need to run to do the scaffolding. And of course, the Vertebrate Genome Project Slack channel. I encourage everyone to join that. And if you have any questions, you can just pop it on Slack. I check it daily. I think Arang checks it pretty frequently as well. So we're there to help out if you have any questions or if you encounter any job failures, anything like that.
Could you hear me?
Yep.
Which page was that again? I'm at GitHub, VGP Assembly.
So this is on VGP Assembly. There's a document called Scaffolding Workflow README, so .md.
Got it. Thank you.
Yep. So to tell you a little bit about the DNA Nexus platform, this is a cloud-based platform. For the VGP projects, you'll be dealing only with AWS US East, so that's where all of the data is stored and all of the analysis is run. AWS has other regions that you can access through the platform, but you generally won't have to worry about that for any VGP projects. And the platform contains projects that we consider to be units of sharing. These are like hard drives on your cluster that you can set permissions for. So some projects might be shared only with my individual username. Some projects might be shared with whole organizations, which are groupings of users. or you can share them individually with people you want to collaborate with on the platform. If I navigate into a project, I'll go into the VGP Tools project. You will see the project contains folders and files, very similar to a file system. It also contains executables. So here we have some workflows. You might also see these objects called applets. These are executables that live inside of a project. And you can also access tools which are called apps by going to this tab in the upper left-hand corner. So these are just different types of executables. If I wanted to organize data in my project, I could navigate to this folder. I could click and drag files into different folders to organize them, just like you would on your local file system. The key difference with files on the DNA Nexus platform is that they exist in an immutable format, so that files, once they're written on the platform, can't be overwritten and can't be modified. The way we manage that is if I click this file info here, you'll see that each file has a unique file ID. That has a full history of how the file was created. In this case, it was uploaded by Sergey. The file name is just a piece of metadata. So you might see some files on the platform that have duplicated names. You can still keep track of which file came from where using the file ID and the history associated with the file. So don't be surprised if you see some files with duplicated names. And then if I navigate into a project that has been linked from the Amazon bucket, you will see that it has this genomic data folder structure that Arang was mentioning. So in this case, we have some ARIMA Hi-C data, some BioNano data, and some PacBio data. And you'll see the files look just like normal files on the platform. So if I open this file info page, I have a unique file ID associated with the file. I have a history of how it was created. And then I also have this details ellipses here. If I click that, you can see it's coming from the container GenomeArk, and here is the AWS S3 path for that file. So this file looks and acts just like a file on the regular DNA Nexus platform, but it's actually pulled from the GenomeArk AWS bucket so that there's no storage charges associated with it. And we'll go a bit more into detail on that later. So in addition to files on the platform, like I mentioned, we do have workflows. So we've streamlined the assembly workflow pretty well. So it's just a single Falcon and Falcon Zip workflow. And then these are all of the scaffolding steps. This will change as we streamline the scaffolding workflow as well. So we'll try and cut it down to a single scaffolding workflow as well as a single assembly workflow. So you might see this change over time, but we'll post updates on the VGP Slack channel and mention them in the group as well. So if I go ahead and open this assembly workflow, this just goes through all of the Falcon and Falcon Unzip steps. It's pretty long, so it takes a minute to load. There's actually 18 steps that are part of this workflow. It starts with the PacBio raw reads, which are usually in BAM format. Then it creates FASTA files out of those, so it just takes the raw reads and converts them to FASTA files. We create what's called the raw read Dazzler database, compute some assembly statistics. Over here, we added a couple of repeat masking steps. So the TAN mask masks tandem repeats. and the rep mask masks global repeats. That's just looking at the k-mer distribution and the raw reads. So if we have, you know, k-mer accounts that are at the 500 level, that's probably a repeat. And then we do overlap of the reads, generate these error-corrected reads. So these are reads that have been error-corrected with the short reads, error-correcting the long reads, and then again run through.
But wait a minute, error correcting with the short reads happens at this stage here?
So in the first half of the Falcon workflow, the error correcting with the short reads happens. We get these consensus long reads that are then used for overlapping the actual assembly.
Maria, you mean short pack bio reads, not
No.
short
Yes,
reads
yes, I mean short pack bio reads.
Short pack bio reads. Okay.
Yeah,
Okay.
sorry
Yeah,
about
the
that.
shorter pack bio
Okay.
reads are used to correct the
Yes.
longer pack
All right.
bio reads.
All
Not
right.
what you're thinking, Eric.
Okay.
So all of this happens only with pack bio reads. We have not added any of the other data types yet. In the second half of the workflow, it actually just repeats what we just saw. So again, it overlaps, creates this Dazzler raw read database now with the error-corrected reads, masks the repeats again on the error-corrected reads, computes overlaps for those, and then the final step is running the actual final Falcon assembly. And then we do it all again with the unzip portion of the pipeline. So again, it tracks which reads came from which contigs, aligns them, phases them, does the haplotype assembly, and then finally does the polish. So this is a pretty long workflow, but as you can see, all of the apps downstream of the first couple of apps have this runnable tag in them. That means all of the inputs and parameters have already been configured for all of these apps. So the only thing I would have to do if I want to run a Falcon or Falcon unzip assembly is provide these input files and configure the parameters of these first two steps. So if I wanted to configure some of the parameters here, I could click this gear icon next to the app name. This will show me the individual parameters for each app. The main one that I will need to configure is the estimated genome size, which I can either get from literature or from running GenomeScope. And then there are some other parameters that can be configured for each app if the assembly doesn't come out very well. But for the first run through, I would encourage everyone to just use the default parameters. And I'll go into a bit more detail about how those parameters are set as well.
I think I should mention something really quick here, because we just learned this internally. The DB split parameter, the minimum read length, oh, that's not there. It can be important to set the split size for a DB split.
Okay.
We end up with a lot more contigs for bacterial genomes if we have too large a split size. Yeah, so we'll have to add that eventually here. We'll talk about that later offline.
Yeah, so some parameters haven't been exposed on these workflows, but as we figure stuff out like that, we can add them as well.
Is this 2000 minimum cutoff is what I was referring to before? Or is that something different? About, you know, when I was asking Arang about the 10,000 versus 1,000 base pair cutoff.
So there's two different cutoffs. Yeah, so there's the first cutoff, which is what is the cutoff for separating the long reads from the short reads for the error correcting step. And then there's the second cutoff, which is which long reads you would use as the primary read in the actual assembly. So this first cutoff is for the error correcting step. If I go down to the second copy of this create raw reads Dazzler DB app, that will have the second cutoff which is for the second step. This cutoff is actually typically automatically determined by the pipeline, but there's a minimum value that we set in case the automatic value in the pipeline is nonsensical. So that's what that minimum does, but we can tweak the formula of how we determine what that cutoff should be for the actual assembly. So that's just a quick overview of what the pipeline looks like for the Falcon and Falcon Unzip assembly. I'm going to go into an example now of how you would actually run it if you just logged onto the platform and wanted to run an assembly yourself. So if I just logged onto the platform, I'll see this projects page. I could navigate into a project that's already been shared with me that already contains the genomic data that has been symlinked from the AWS bucket, or I could create a new project for my test assembly. So I'm just going to call it my test assembly. I can set the billing account to any org that has been shared with me that allows billing. So my user account typically doesn't have a billing profile associated with it. You get about $50 of free credit to play around with, but typically assemblies cost a lot more than that. And so you want to set the account that has been set with you, shared with you. The org Eric Lab account is where we run all of our production assemblies. That has significant discounting on the compute cost because of the contract that DNA Nexus negotiated with VGP. But the bill goes to Eric, so that should only be used for production orgs. And then the Myers Lab has kindly offered their credits for the purposes of training this group. So all of you should be added to the Myers Lab org account where you can run your test assembly. And that has some compute credits where you can run a test assembly. So I'm going to set it to the Myers Lab org. Yep.
What is the average price of a mammalian assembly?
I do not have that number off of the top of my head. Without the VGP discount, I think it's around 1,000 or 2,000 for a reasonable size mammalian assembly, like human or smaller. With the VGP discount, it's about 60–70% lower than that.
Well, it's been coming out higher than that for the Falcon Unzip. At least the Falcon Unzip is the most expensive part in terms of compute, and everything else is much less.
Right.
Yeah, I think the mammalian ones without the discount are more like 8,000 to 10,000.
Right, and then width could be down to 2,000 or 3,000.
Right.
Yeah.
Yeah.
It's likely proportional to the genome size.
Yeah.
That's right. So assembly is a quadratic problem, so it scales quadratically due to genome size because of the overlaps that need to be computed. So it can get very expensive very quickly. So we encourage everyone to be very careful with the funds here.
And if you don't use the mass repeats, it takes even longer.
That's right.
Which is a worry of mine technically, but it certainly saves money.
So if I go ahead and create this test assembly project, I can click this add data button to add some data to it, including apps and workflows that I want to run. So I can upload data directly from my computer or from a server. Keep in mind that this data wouldn't be symlinked to the Amazon bucket, so you would have to pay storage for hosting that data on the DNA Nexus platform. I can also copy in data from another project that's been shared with me. So all of you should have access to this Arabidopsis assembly demo project, which has some Arabidopsis PacBio raw reads to do a demo assembly. This assembly, I ran it earlier. It's on the order of $200 without the discount. So you can't do too much damage by running this one. And here I can select an entire directory that has some input files. So these are just some PacBio raw reads. So I'm gonna go ahead and click this folder, add it to my project. And now I have some raw reads that I can do an assembly with in my project. I also want to copy in the assembly pipeline. So clicking again this Add Data button, I'm going to go to this Other Project tab and navigate to VGP Tools and select the Falcon and Falcon Unzip assembly workflow. So this copies in the pipeline so that I can run it in my project. You'll see that some projects are shared with you with different levels of permissions. So if you wanted to add a collaborator to this project, you could click this share button and select a member to add. For example, if I wanted to add all of Eric Lab, I could select the org name, Eric Lab, and then I can set the different levels of permissions to add that user to. So a viewer can only view and download data. Most of you are viewers on, for example, the VGP Tools project, so you're not able to run any analysis in that project, but you're able to copy data from that project, including the tools. An uploader can upload data, but not edit the data, rearrange it, run analyses, anything like that. So this would be someone like a sequencing service provider who's uploading data to your project, but can't actually do anything in the project other than add data to it. A contributor can manage the data and run analyses, but can't set user permissions. Yep,
Yeah.
Was there a question?
For the uploader, when they upload the wrong file, can they delete it?
They cannot.
Okay, just so we know, thanks.
Yep. So a contributor can manage the data, run analyses. Typically, they would be able to delete data, though you can set a special permission for that so that only administrators can delete data. But typically, they can do most things in a project that an administrator can. The only difference is that they cannot change user permissions. So they would not be able to change access permissions for any of the users that the project has been shared with. So I'm going to not add anyone to my project for now, but I've already shared with you a project that has a pipeline example in it. So we'll take a look at that one in a minute. So once I have my workflow and my inputs in my project, I'm gonna load the workflow run analysis screen again. This is what we were just looking at a minute ago. But this time, since I have permissions to run jobs in this project, I'll actually be able to run it once I configure the inputs. So for most assemblies, the only inputs you will need to configure is add in the raw PacBio reads. So I can select those here in my project. And then I will also want to configure the genome size just in this first Create Raw Reads Dazzler app. So if I set this gear icon, I can set the estimated genome size here. And for Arabidopsis, I believe it's around 135 megs. I could be wrong, but this is something you would typically look up or run GenomeScope to find out. So once I save that, I should see that all of my apps are now configured.
Maria,
Yep.
just so I noticed that the numbers before were like, you know, real whole numbers, and here you type an abbreviated one, 135M.
Mm-hmm.
So it's recognizing M as megabases?
Yes.
And
So
what about G and B, like for gigabases?
Yeah, so there's 3 valid prefixes. There's a lowercase k for kilobases, a uppercase M for megabases, and uppercase G for gigabases.
Okay.
And you could do 1.5 G for 1.5 gigabase genome. So it recognizes decimals as well.
All right, thanks.
This is just to make sure if you're running a very large genome, maybe you miscount a number of zeros, and then your assembly wouldn't turn out too well. So just for convenience, it does recognize those prefixes. So if I go ahead and save that, you'll see that all of the apps are now configured, and I could actually click Run as Analysis to launch this workflow and run all of the steps in the Falcon and Falcon Unzip assembly. One thing that I would encourage everyone to do to keep their data organized is also configure an output directory for all of the workflow outputs. And so you can do that by clicking this workflow actions and setting the set output folder option. So I can create a folder here. That's my assembly intermediates folder. And then select that as my output folder. All of these apps, if you open one of the app settings, you'll see they also have an output folder configured, so that will be a subfolder of the folder I just specified. The other things you can change, which might come in handy if you're doing troubleshooting, is you can change the app version. So sometimes we'll have a failure that happened due to a bug, and we'll update the version of the app. So you can load the new version here by changing the app version in the workflow. And sometimes you can also change the instance type. So if you have an unusually large genome, you might want to set a bigger instance type by selecting the instance type here. So the higher instance types cost more, and lower instance types cost less. And so we do want to use large enough instance types to fit the genome, but not so large that we're paying extra. So most of these have already been configured for the assembly. So you typically wouldn't have to change it unless there's a failure due to insufficient storage or insufficient memory.
Maria, can I ask something?
Yep.
So the payment is for process, because I have an experience with like open up Amazon machines that you kind of pay for the hours that it's open. If you're using it. But in here, if we run Falcon and Falcon finishes, then it stops, or it's still running for the prices?
So the payment is for compute, but it's only for compute that you actually use. So we have a lot of smart job managers on the back end that open up the Amazon workers and then shut them down when the process finishes running. So you're not paying for having a worker open that you forgot to close, but you are paying for the compute time that the worker was open.
Great, thanks.
So then this difference in the instance type really then is going to affect how fast your assembly is generated.
That's correct.
So if you take the more expensive one, is because maybe you want your assembly done in 1 day as opposed to 4 days or 5 days.
That's right. And then there's a trade-off in terms of, do I want to have a more expensive instance open for a shorter amount of time? And is that going to be cheaper than having a cheaper instance open for a longer amount of time? So there's some tweaking there in terms of optimizing for speed or for cost.
How about for the jobs that fail? Let's say you choose an instance that's too small to run the final unzipping stage. What will happen?
So if you run a job that fails, you actually still have to pay for the compute that was used by that job because DNA Nexus gets a bill from Amazon, so we have to pay it. And so for most cases we'll want to configure the app such that jobs fail as infrequently as possible.
Maria, I think we have to be a little bit flexible. If not in this particular case where you choose the wrong instance type, but like if a job fails because something didn't work on the DNA Nexus platform as was expected to.
Yep.
Yeah.
Yeah, so there's always going to be exceptions there. And if a job failed and you have absolutely no idea why, you can ping me on the VGP chat group. I'll investigate it. If it's found to be due to a bug in our apps or a platform failure, that money will be refunded. But that's going to be on a case-by-case basis.
Yeah.
So once I've configured my apps, I'm going to just click this Run Analysis button. That's going to take me to the Monitor tab in the same, I must have configured something. Oh. So you might see this error when you try to run it. That's because one of the instance types that's configured for this workflow has not been set for this org that I set as my billing account. So I'm just gonna go down here and change that very quickly. I think it was here. It was one of these last either assembly or polish steps that had this extra large worker. I'll troubleshoot that in a little bit so that all of you can run a test assembly in your own project.
Well, nothing better than seeing some things that need to be carefully chosen in real time.
Right. So we've chosen some large instances because we had a lot of assemblies that were failing, specifically in those steps because they require a lot of memory. So we'll have to make sure that's configured for the billing account for the assemblies that we actually run. But for the magic of TV presentations, I already have a project where I've run an assembly, so I'm just gonna navigate to that. So this should be shared with all of you. It's this Arabidopsis assembly demo project. You can see I've done exactly the same thing here. I've copied in the inputs, I've copied in the workflow. And then when you click Run Analysis on your workflow, it will actually take you over to this Monitor tab inside of your project. So this is where you can monitor any jobs that are running or have run in your project. If you have permissions to view billing, you'll see the price here. This is not using the VGP discount. So depending on your billing account, like I mentioned, only Eric Lab will have the VGP discount pricing. And then if I click this Falcon and Falcon Unzip assembly workflow, this is my analysis that has already finished. So just like files on the platform, job histories are immutable. So you can't delete a job history without also deleting the project. So you always have a history of exactly what was run with which parameters, how long it took, how much it cost, stuff like that. And the output files are linked to that analysis as well. So if you look at the Falcon workflow, again, we have the same steps that we saw in the run analysis screen. So we have our BAM to FASTA conversion, we have our raw reads, Dazzler database creation, the repeat masking, overlaps, and then all of that repeated again. And you can see by the green line here how long it took in wall clock time. A lot of these are configured to spin up many workers to finish the assembly faster. So it runs everything in parallel. So if I click this D-aligner job, I'll see all of the workers that actually launched to run this app. So you can see it's probably about 100 workers that it ran. And it already configured all of the logic for how to split up files across workers so that it runs efficiently. If I click on one of these tiny jobs, you can see the exact parameters that were used to run D-Aligner. This is something that you can configure, but I would really encourage you not to, because it can dramatically change how long your assembly takes. And you can see the exact input that went into that job and the output that was created. If you see this file ID, that means an intermediate output has been deleted because it's already got used up in another subjob. And then the other thing I can see for each single job that I run on the platform is the exact log. So if I click this view log button, this will actually show me the standard out, standard error, and information for what happened on the Amazon worker that I ran. So info just gives you resource utilization information. So you can see an example of that up here, where it shows you roughly every 10 minutes how much CPU you're utilizing, how much memory you're utilizing, how much storage you have available. And then you can see the actual commands that were executed on the worker. So the first thing it has to do when it spins up an Amazon worker is unpack all of the resources that are associated with this job. Those would be any binaries, executables, or dependencies for this job, as well as your actual input files that live in the Amazon storage bucket have to be downloaded onto the worker in order for the job to process. So you'll see it downloads this file here using our DX command line utilities. Then you can see the exact command line that was run. In this case, it's running D-Aligner with all of these pre-configured parameters. This is the log that comes out of D-Aligner. So you can see all of the information here. And then at the very end, it tars up all of the files that are produced and uploads them back to the platform. So in addition to downloading files onto the worker to process them, you have to upload the files back into storage for them to show up as an output on the platform. Does anyone have any questions about jobs and job logs?
Yeah, one quick question. I'm a little lost. How did you get this? Go back one on your screen.
Here?
I mean, close the D-Aligner thing.
Yeah.
This. Go back one more. You had a graph that had the whole workflow and runtimes?
Oh, yep. So I went to the monitor page. This is a workflow I had already run in the past. So if I click the workflow here, I'll see all of the apps that were run. If I click an individual app, I will see all of the subjobs that it ran, so any child workers that it spun up. And then if I want to go back to the parent analysis, there's this parent execution ID. I can click that. It will take me back to the full workflow.
Wait. Where's parent execution ID?
So if I click a subjob here, if the subjob has a parent, it will have this link here where you can navigate back.
Okay, great. I see it all online. Thanks.
Great. So if I go back to the parent analysis, I wanted to go through a bit of the outputs that are produced by Falcon Unzip.
Yeah, I have a question.
Yep.
So you went through these logs and it has been written about the resources that this specific process have been using. Do we use this information to optimize our resources usage?
So we as app developers use it to optimize the resource usage when we configure the apps. Typically, when you're just running a workflow, you wouldn't have to use that information except for troubleshooting purposes. So if your workflow failed halfway through, you might want to go look at the log and see what caused the failure. Any other questions? Okay. So I just want to point out a couple of outputs that are used to configure the parameters in this workflow. So the first thing we do before actually running the assembly is we do this pre-assembly calculate read length distribution graph. So if I click this job, I can see this is using just the raw reads, not even overlapped reads, and the genome size that I expect to see. And it bins the raw read lengths down here into coverage bins. So for this first bin, this is the cutoff that I would need to set if I wanted my assembly to only use 10-fold coverage of reads for the genome size that I provided. So I could throw out all of the shorter length reads and I would still have 10-fold coverage of my genome. And then it goes up incrementally from there. So this is 10-fold coverage, 20-fold coverage, 30-fold coverage, and so on, until you get to a too short of a read length so that you wouldn't be able to cover your genome anymore. So based on the number of bins here, you can see how much fold coverage you have of your genome. So in this case, it would be about 60 or 70-fold coverage for the raw reads that I provided. And this is actually what's used to configure the length cutoff. So typically we'll take something around 50 or 60-fold to do the overlap reads, and then from the overlap reads we'll want to do something around 20 or 30-fold to do the actual assembly. The pipeline is already configured to compute those parameters automatically. So if I go back to the parent pipeline, I believe it's used in the D-aligner step. One of the inputs that D-aligner receives is the length cutoff for D-aligner. So this is the length cutoff that is automatically generated by the workflow. If you wanted to change that length cutoff, and this is what Eric was asking about earlier, you could actually configure that input before running the workflow to use a manual length cutoff instead of automatically generating it. I would encourage you not to do that for the first time of running an assembly, but that's one of the parameters that you can tweak when you rerun an assembly or want to manually set the parameters for your assembly. You can also set these more advanced command line parameters for the aligner. So this is stuff like your K-mer size and your seed size for actually computing the overlaps. And again, these are parameters that could dramatically change the runtime of your assembly. So they could cause your assembly to cost a lot more than you expected. And then for the outputs, if I go back to the Manage tab here, that'll take me back to my file viewer screen. This assembly is already complete, and I had it run. I sent the output into this outputs folder by configuring the output folder in the workflow screen, as we did earlier. And you'll see the Falcon Unzip creates these individual directories for each of the steps in the pipeline. So if I click the BAM to FASTA step, I should just see my output FASTA files that were converted from my raw BAM files. And then each stage of the Falcon workflow creates its own outputs. So stage 0 is the calculate read length distribution. Stage 1 is my, I believe, raw read overlaps. And then stage 2 is my error-corrected read overlaps. Stage 3 is the actual Falcon assembly, not Falcon Unzip yet. So when you run a Falcon assembly, you'll get this p-config FASTA file and this a-config FASTA file. That stands for primary config and alternate config. And then Unzip uses those to actually separate the primary from the haplotype. So if I go down to my final output from Unzip stage 5, I will see this p-config FASTA file that's my primary contigs, and H contig, that's the haplotigs, so the alternate haplotype. And you'll see FASTQ files that contains the exact same information, just in FASTQ format. So these are the outputs I would then use for scaffolding the workflow from here. Any questions so far? One more thing I wanted to show about running Falcon and Falcon on Zip is the Jellyfish and Genome Scope app. So this is, there might be a copy in the VGP Tools project, but you can also navigate to this Apps tab at the top left of your screen. Here you'll see all of the public tools that we've made available to all users of the platform, and also VGP-specific tools that have been shared with you individually that have been built as apps. So if I wanted to search for a specific tool category, I could, for example, look for all assembly tools that have been shared with me, and you'll see all of the Falcon apps that have been made available to me. You'll see a copy of the Canoe app that we have available for bacterial genomes. And then you can also search for apps by name. So if I wanted to run Jellyfish or Genome Scope, I could run search for that app by name. And we have this Jellyfish and Genome Scope app. This is the app that you would run in order to estimate your genome size based on the 10x FASTQ files, so your Illumina raw reads. When you first run an app, you might see this install button. All that means is you're agreeing to the license terms of the apps. So you can see, usually you can see license information here, or you can navigate to the upstream project to see the license associated with the app. Most of the apps are open source, so it's not something we typically have to worry about. So if I go ahead and install the app, that just says I'm agreeing to the license terms for Jellyfish and Genome Scope. That's just, you know, standard, I think, MIT3 license. And then if I want to run this app, I can select the project to run it in. And you'll see, again, this run analysis screen that we saw earlier with the workflow to run this specific app. So all I would have to do is provide my Illumina raw reads, and I believe this also takes in a genome size. Well, no, it doesn't, because we're using that to estimate the genome size. So just provide the Illumina raw reads, and you can set the k-mer parameters. Any questions about Genome Scope? Okay.
I have a general question.
Yep.
K-mer size, is there some presetting, or would be basically, I know for Jellyfish normally you test like different K-mers, like 31 or go even a bit higher?
Yeah, so we have a default setting that's configured. This is a very cheap app to run. And so you can test different k-mer lengths for your specific genome.
Arang, do you have a comment on what the typical k-mer size is that you find helpful for Genome Scope?
I wouldn't use anything below 21 mer for a 3 gig genome size, because you're just going to have everything be repetitive. But typically we use between 21 and 31 probably.
Yeah, for Illumina reads, yes. For PacBio raw reads, you might lower the K length. For corrected reads, probably 18 to 21.
Yeah, for PacBio raw reads, you're not going to get a prediction anyway.
Right.
Okay, good. So we should try basically between 21 and 31. Or we could also start with the 18 default, or should we set that to 21 at least?
For, I mean, if your genome is smaller, like if it's a bird genome that's a gig, 18 is probably okay to start with. If you're looking at a mammalian genome, there's not enough 18 mers that you're going to start having random collisions where, you know, you'll just have k-mers collide by chance in a random string that long. And so it might end up giving you a worse prediction because of that.
Okay.
Like more of your genome will look repetitive and it'll have a harder time fitting the distribution.
Okay, thanks.
So now I wanted to go over some of the scaffolding apps. So I'm going to be referring to this VGP README that I added just to run through each of the steps. I've tried to make it pretty easy to follow. Obviously, it could be improved. And I've shared with you all a project that contains Drosophila files from all the different data types based on Sergey's suggestion. So this isn't exactly how the data will appear when it's uploaded to the VGP bucket, but it's pretty close. It's all the same data types, and you'll see there's some changes in file names and stuff like that because I pulled them from different published papers. So if I wanted to run a scaffolding workflow after my Falcon and Falcon unzip assembly finished, again, I would probably use the same project that I had before with my test assembly. And then based on the inputs, I would add the appropriate scaffolding steps that I need to use for this particular genome. What I mean by that is if you go into the VGP tools project and add data from it, you have a few choices that you need to make. So based on whether you expect your genome to actually be diploid or not, you might want to skip the purge haplotigs. I would really encourage everyone to run purge haplotigs part 1, which gives you the distribution, which I'll go into a second. But if it's not diploid, then you can skip purge haplotigs part 2. And then based on what bionano data is available, if there's bionano data available from 2 enzymes, you can use this scaffold 3 2-enzyme workflow, or if there's data available from DLE or only 1 enzyme, you can use this 1-enzyme workflow. And then you would add in steps 4, 5, and 6 as they appear. If your genome is bigger than 4 gigabases, this step 5 will have a lot of issues. So I'm going to be adding another step 5 option that has Minimap aligner instead of Blazor aligner, since Blazor doesn't accept genome sizes that are bigger than 4 gigabases. So we're gonna do some tweaking of these steps, and you'll have to just look at the data that you have and choose the appropriate workflows. And I've outlined all of that in this step-by-step. So here for purge haplotigs, after running step 1, you'll have to manually determine, do you need step 2? And if you do, what are the thresholds? And then when you're running arrow polishing, you'll have to manually determine, am I running arrow polishing with Blazor for genomes less than 4 gigabases, or am I using Minimap for genomes greater than 4 gigabases? So once I've added in the appropriate workflows, I can start filling them in with my data type. And we're doing sequential scaffolding, so you can't just fill in all the workflows and then run them all at once. You're actually using the outputs from the first workflow to inform the inputs of the second workflow. When you copy in the workflows, you might see there's some applets that got copied over. It doesn't matter where they are in your project. I like to just create a new folder and organize them just for convenience. Most of these will get transported into apps, which we were looking at earlier, so you won't see them get copied into your project at all. So assuming I have my Falcon and Falcon unzip output, I'm going to run this first purge haplotigs step. And I'll just take a second to load. And this step consists of 4 apps that have been configured. The very first app is just this rename contigs app. And all that does is it looks at your FASTA file input, and sometimes the last step of unzip, which is the arrow polishing, will add in this pipe character and the word quiver at the end of the contig names. Purge haplotigs doesn't like that. It treats it as a bash pipe. And so that could cause a failure. So we just remove that pipe character using this rename contigs app. So here I would supply the FASTA file that is the output, the final primary contigs output from my unzip assembly. And then purge haplotigs uses the raw PacBio BAM files or FASTA files to do a read coverage histogram. So I can supply the reference FASTA. That's going to be my assembly output from unzip stage 5, and only the primary contigs output. And then for the raw reads, these are going to be in FASTA format. So again, if I go to my Falcon and Falcon unzip output, I can go to the BAM to FASTA output, where my files have already been converted to FASTA format, select all of those, and provide those as the input for my raw reads. I'm just going to upgrade the app version here. You won't have to do this because the workflow should already be configured for you, but I updated it earlier this morning. And so now all of the apps in the workflow are runnable. Again, I would encourage you to set an output folder for your analysis. So I might put it under assembly intermediates, or I could add a scaffold intermediates folder. And all of the outputs of this workflow will go in that folder. If I click Run as Analysis, what it's going to do is take all of the PacBio raw reads and align them to my primary contigs, and then look at the read coverage of the read coverage depth of how the reads align to the contigs. So if you go to the Purge Haplotigs tutorial, you'll see what we expect to see is something like this. So you'll see an output PDF as the final step of the workflow, and it'll look something like this, which is a histogram of read depth along the size of the genome. And so if your primary contigs should have pretty high read depth, which will be this large peak, but then you'll also see some of the reads mapping to the alternate contigs with an unequal depth. So these are the alternate contigs that we want to purge in our Purge Haplotigs workflow. And you can use the second peak to determine the cutoffs for that purge haplotype step. So you'll use the midpoint of the peak as your midpoint cutoff, and then somewhere around here as your low cutoff, and somewhere around here as your high cutoff. So this is something that you'll have to manually configure. If you only see a single peak as your output, then that means there are no haplotags in your assembly, so you don't have to run the purge step, and you should be good to go.
This is Eric. I'm back. I'm saying I think it's a very good idea just to do it anyway, just in case there are a few haplotags here and there that need to be purged, because even if there's 1, 2, or 10 genes with artificial duplications, that could get reported in literature as an error.
So we'll have to set some guidelines as to how to determine the cutoffs if there's only one peak.
So Maria,
maybe based on the long tail.
I have a script put on the pipeline in the pipeline purge haplotics that determines the cutoff.
Oh, okay.
It basically gets the histogram as input and tries to de-differentiate where the boundaries are. But it's not a perfect script. You still have to, I would still encourage people to look at the histogram in case if the genome is not so highly divergent, you won't be able to see the first peak for some cases. Yeah, I guess other than that, I have tested the script works on at least for the first 17 genomes. I just had 2 exceptions where there were clearly just one peak. So yeah, other than that, yeah. Yes.
So we can update the app to output recommended cutoffs as well as the histogram. So once I have my cutoffs, I'm going to go back to my project and run the part 2 purge haplotigs step. And this is just taking in the primary contig reference genome, the alignment file that was produced in the first workflow, the genome coverage file that was produced in the first workflow, and then I can use this gear icon to set my low, mid, and high cutoffs for configuring the purging of the haplotigs. And then as output, it will give you some coverage statistics as well as your curated assembly and your curated haplotigs. So this curated assembly is what I will be using to do the downstream scaffolding up until we recombine the assembly with the haplotigs. Any questions so far?
I have a general question, which maybe dates a bit back. So even if the genome is not diploid, we're supposed to run purge haplotics, at least of part 1 anyways, right? Just for my understanding, why should we run it as well?
We assume that your genome is diploid.
Okay. So
yeah.
just for safety reasons, basically. Okay.
Well, yes. Well, assume it's not only diploid, but I mean, if it's not diploid, then maybe it's not even necessary to run purge haplotigs.
Right.
But the reason for purge haplotigs is that the Falcon Unzip doesn't fully get rid of the haplotypes. So we need this extra step to do that.
Okay.
I would imagine there could also be a very large repeat that gets mistaken for a haplotig. Right. So it may be better to purge those as well. But yeah, this step would be optional for a non-diploid genome.
Sorry, okay.
Yeah, no, go ahead.
No, it was basically just, even if it's not generally run it anyways,
Yeah,
just to make sure that there's.
Mm-hmm.
But Maria, if we don't want, if it's a large repeat that belongs in the primary haplotype, technically we don't want that in the haplotags because it's not a haplotype. It's actually part of the primary assembly.
Right.
Yeah.
So the next step of the scaffolding workflow uses the 10x genomics reads to scaffold. So for this, we're using the SCAF10x app, and we run it twice using the same raw reads. So for this input, you would specify your primary contigs, so your curated primary contigs that came out of purge haplotigs. For read 1 and read 2 FASTQ files, you would specify your 10x genomics raw reads. So if they were uploaded to my, I'm just using this as an example project, I would, well, this one doesn't have 10x genomics. I would add my genomic data, 10x genomics, and you'll see some FASTQ files here. If the file name doesn't match, which it doesn't with these Drosophila files I pulled, you can disable the filter by clicking this filter here. And then you'll see that you do actually have FASTQ files in that folder, and you can select those for running your 10x genomics scaffolding. Once I've filled those in, it's going to automatically run this app twice. The first time, if I click the parameters here, it's going to run it as raw input. And you can see the alignment options are already pre-configured for you. In our current workflow, we don't typically run break 10x, which breaks the scaffolds. We only run scaf 10x, but that could change in the future. And you can also set an output prefix for your output files. So I believe this would be like a S2 round 1 output if I'm following the naming convention for the output genomes. And then this produces these barcode reads that have been pre-processed, and those are used as input for the second round of SCAF10x, just so that pre-processing doesn't happen again. So I've already linked the outputs here to the inputs here, and the only difference here is that now we have this raw parameter set to false, and we can set another prefix based on our naming convention. And then we can also set an output folder for this workflow so that all of the outputs would be grouped in the SCAF10x output folder. So once I run that, that'll run the 2 rounds of SCAF10x automatically for me. And I will get this scaffold output, which I'll use in the next step. Any questions about 10x scaffolding? Okay. So if I go back to my handy cheat sheet here, we've now gone through purge haplotigs and SCAF10x. We have our scaffolded FASTA output from SCAF10x, and we're going to use that now in our BioNano hybrid assembly scaffolding. So this step uses the BioNano C maps. These are assembled optical maps produced by BioNano, and they could come as a single enzyme, usually DRE, which is their latest protocol, or some of the older files you'll see will have 2 enzymes, and these are in the CMAP file format, which is not a file format that you see very frequently. That's just the file format for the BioNano optical maps. So in this case, I have my CMAP file from my Bionano optical map. I have my FASTA file. This is the SCAF10X output. I can also provide the arguments file that was used to assemble the CMAP if I have it, or an arguments file to set custom parameters for the actual scaffolding. So if I navigate to my other project here, I can select the CMAP file. I actually got 5 copies because this paper had 5 different genomes in it for Drosophila. But if I navigate to one of them, you'll see it has this refined final CMAP. You'll want to know which enzyme was used to produce it because you'll want to specify that as an input. And then here I would provide my SCAF10X scaffolded output. And under my parameters, I can set filter for conflict resolutions. Usually I use the default parameters, but you can tweak those a little bit as well. And one thing to notice with the Bionano apps is they tend to use a lot of memory, especially for larger genomes. And the thing to be very careful about is that if they run out of memory, instead of crashing.
We just lost audio.
Oh.
Yeah, Maria, we lost your connection.
Can you hear me now? Hello?
Oh.
Okay, you're back.
Okay. Sorry about that. Did you hear me talk about the memory requirements of these apps?
No, we just heard that usually it requires large memory for large genomes.
Okay. Yeah, so these apps usually require very large memory for larger genomes, and they'll actually crash instead of, or they'll actually keep running instead of crashing when they run out of memory. So we've set a timeout for these apps so that they can't run more than, I believe, 24 hours. So they might fail just from reaching that timeout, but the actual reason is that it ran out of memory and silently kept spinning instead of crashing out. So we're actually out of time for today's training. So I apologize for that. I want to very quickly, just in the last 2 minutes, go through the last couple steps. So after Bionano scaffolding, we'll run the Salsa scaffolding much in the same way, but now supplying the Hi-C raw reads. That pipeline is under VGP tools and has already been configured. And then aero polishing and freebase polishing. So those pipelines are also available under VGP tools. And if you have further questions, you're welcome to ask them in the Slack chat. I'd love to take any last questions now.
I do have one real quick important question. I'm a little nervous on how to start something and avoid accidentally being charged a couple $100 personally. Where is it a matter of what project I run in?
Yes. So when you create a project or when you are in your project, you can set the billing account. So if I create a new project, I would set a billing account here to determine who is responsible for any compute or storage charges that I generate. If I have an existing project, I can go to the project settings page and set the billing account here. For running these test assemblies, I would encourage you to use the Myers Lab billing account, since they were kind enough to offer their credits for the testing of for the training of the assembly pipeline. Any other final questions?
So we are basically running the training on the credits. So we won't get actually paying by running anything here. But the credit is not unlimited. So that's why we wanted to start with some small genomes. I encourage everyone to try to build the assembly with the Drosophila that Maria shared.
Okay, good.
I just uploaded our super small genome. We can discuss it in Slack. It's a synthetic genome. It might help to save some money.
Okay.
That's also nice. The question here is, does it have all the 4 platforms or just?
No, you'll only be able to run through Unzip, but at least you can see that much.
I see.
Yeah, so for Unzip, we have a Arabidopsis as well. And I see there's another quick question on genome size. For diploid species, is the genome size based on the whole genome size or the haplotype size?
It's the haplotype size. Yeah.
And
So,
sorry,
what-
just if we understood this correctly. So each one should make a new test assembly or project and basically copy from the Drosophila demo. Is that how it is?
And
So
build
you-
the
And build
So
the-
you're all shared with viewer permissions on the demo project, so you won't be able to run anything in those projects, but if you create a new project, you'll be able to run things there.
Okay, great.
Wait, if I create a new project, that's when we get charged, right?
Yes, but you can set the billing account to the Myers Lab account,
Oh,
which
okay.
we're using for the credit. Any other questions?
So I have my last comments if there are no questions.
Yep.
Yeah, so I just sent an invitation from our VGP Slack channel. So I made a channel called training. If you are able to come into our VGP workspace, then you can find that either under channels or just Slack me or Marcella or anyone you might who looks familiar to you and ask to let you join the channel. And yeah, just write anything that comes up if you think you are in trouble. Yeah, it's a fast way to communicate, and the other people might have the same question as you have. So don't be afraid to throw anything there.
And if you encounter any failures of the workflow, I'm available to help you troubleshoot anything that you might encounter.
Thanks, Maria and Rank. Okay, good.
Okay.